Good morning, everybody, and welcome to the Minneapolis Heart Institute Foundation Signature Grand Rounds of the Year. It's the annual Howard Burchell Lecture. And I say a few words about Howard, and then I'll have Dr. Zashiri introduce our speaker this morning. This is my favorite picture of Dr. Burchell. Perhaps not many in this room, maybe Jay and I and Randy remember Dr. Burchell. Howard was a remarkable physician. He was a scientist. He was a gifted teacher. And above all, he was a mentor and spent time with each and every one of us. And I still have diagrams of his writing on papers that I wanted to submit and his critiques of them, which were quite humbling at the time. Howard had this beautiful shock of white hair and a very gravelly voice. If you ever heard it, you'd never forget it. And he would always spend time with you. Howard was actually from Canada and from Ontario, I believe, and spent a lot of his early career at the Mayo Clinic. And in fact, he was with Dirk Durer, was one of the pioneers in the field of electrophysiology, appropriate to our talk today. I lifted this diagram that Howard made. This is a map of a patient with WPW in 1967 at the time of surgery for a repair of an atrial septal defect. And Howard, at the time of surgery, mapped out the earliest activation site, which you see down here at the infralateral aspect of the right heart. And with Dr. Dwight Magoon at the Mayo Clinic, during the surgery, they injected lidocaine at that site and were able to block the conduction of the accessory pathway. Now, this was not a permanent ablation, so once the lidocaine wore off, but I thought it was an elegant way of proving that the connection could be interrupted. And then ultimately, now we have a catheter ablation systems to do this. But Howard was at the forefront of this, and he always loved digitalis. It was one of his favorite drugs, and he grew foxglove plants at his home. He was the editor of Circulation, one of the earliest editors in 1970s, and was that editor for 4 years. He was also the chair of cardiovascular medicine at the University of Minnesota. And when Howard retired from that position, we adopted him here at this institution. He spent the last 25 or 30 years of his career here. And if you ever wanted to find him, he'd either be here at Grand Rounds or he'd be in the library, one of the two places. We have a distinguished list of Burchell lectures. Howard passed away in 2009 at the age of 101. And we started this in 2010. Dr. Bernard Gersh from the Mayo Clinic was our first. And our most recent was Dr. Duke Cameron, who last year presented on Marfan syndrome and surgery for this. And so it's appropriate, I think, in this distinguished list to have Dr. Zashiri introduce our morning speaker. So Edwin, please.
Thank you all for coming for Grand Rounds today. So it is indeed my pleasure to introduce Dr. Chung to the audience today. So Dr. Chung is a clinical cardiac electrophysiologist and a translational research scientist at the Cleveland Clinic main campus in Cleveland, Ohio. She aims to accelerate bringing basic science advances and discoveries from arrhythmia research back to the bedside. After her early work discovering the cytokines interleukin-1 and TNF-alpha and the ability to inhibit contractility in cardiac myocytes, and earlier work of discovering the role of inflammation in atrial fibrillation, she and her team have contributed to biorepositories of AF genetic discoveries and made functional and causative linkages for several AF risk loci, and applied induced pluripotential stem cell derived cardiomyocyte and AI approaches to multiomics for therapeutic discovery. She has had a distinguished academic career and led multiple AHA and NIH sponsored clinical projects. She is the leader of a program project study on AF that explores new approaches to accelerate therapies for AF and genetics. Dr. Chung has also chaired the 2020 AHA scientific statement on lifestyle and risk factor modification for AF, and the 2023 Heart Rhythm Society guideline on cardiac physiologic pacing, which is very topical now. She also served as vice chair for the 2023 ACC-AHA ACCP Heart Rhythm Society AF guideline document. She has served as the chair of the AHA ECG Arrhythmia and Data Science and Precision Medicine committees. She is a clinical science lead for the AHA committee on scientific programs and served as director of the AHA COVID-19 Research Grants Coordinating Center. She is president-elect of the Heart Rhythm Society, and she has been named as one of the best doctors in America since 2021, and she has been able to get that every single year. She has received numerous awards, including the Distinguished Cleveland Clinic Research Mentor Award, Clinical Research Award, and Excellence in Clinical Research Awards, as well as the AHA Bringing Science to Life Award and the AHA Clinical Cardiology Council Distinguished Achievement Award. I've had the privilege of knowing Dr. Chung since the summer of 2008 when I was a clinical cardiology fellow at Cleveland Clinic, and over those years, she has mentored me. We've had the opportunity to write some papers together. I've been inspired by how she has a passion for taking care of patients, teaching, research, leadership, and innovation. I'm absolutely delighted that you have agreed to come and give the 2025 virtual lecture. Mina, thank you.
Wow, thank you for that introduction. And this huge honor of being able to give the virtual lecture. I mean, these people are just amazing that you've invited, and it's really such an honor for me to be here to talk with you. And you know, it's I don't know. I would love to meet with each of you individually to talk what we can do for our field. And I've just been really fortunate, I think, to be able to be in a position to be able to help. And I'm not sure that how much we've actually accomplished, but you'll see through this talk that sometimes we take some setbacks, and we also want to kind of take advantage of that and move forward. So this topic that I have is looking at AFib genomics from bedside to bench and back again. And you know, it really intrigued me that Howard Burchell had some background in cardiac electrophysiology, and you've already heard a lot of this. I would call him a Renaissance cardiologist. You know, he was so good at so many things in so many fields, and besides what you heard about Mayo Clinic and what he did at University of Minnesota and being editor-in-chief of Circulation. I love that picture too. You know, there's this, as you probably know, there's this nice paper in American Journal of Cardiology in 2010 where various people had personal reminiscences about him. And I was especially intrigued when I saw that he helped to launch modern interventional EP with his work in WPW and this temporary ablation and of WPW of a Kent pathway. So what a, you know, he just was a forward-thinking person, and so that was really amazing. And so these quotes that were in this article were really interesting, and I thought one of the quotes that I really enjoyed was, "How can you call atrial fibrillation the grandfather of cardiac arrhythmias? Does celibacy protect against atrial fibrillation?" Well, that's especially relevant to my topic today because, you know, now that we're learning that there's could be a genetic basis to atrial fibrillation, yes, maybe celibacy could keep you from passing it on. But anyway, so the name of my talk is taking AF genomics insights and going from bedside to bench and back again. And I think that's what we've sort of done in atrial fibrillation. And we've gone from learning from our patients to taking, that what we've learned in genetics from them to the bench and then hopefully back again. So this is an outline of what I'll go through over the next few minutes. We're going to talk a little bit about AF epidemiology and pathophysiology, then what we've learned about AF genetics and genomics and its association as well with structural variations, then the age paradox we'll talk about, and then some of the pathways we're taking towards a genomic-based drug discovery. We've also been looking at imaging and trying to look at genomics for interventional targeting. I left that out just to leave ourselves some time for questions, but happy to talk about that as well. I want to start off by telling you, if you don't already know, I'm sure you do know how important atrial fibrillation is. So this is a slide that gives some updated statistics on atrial fibrillation from our AF guideline. So as you know, prevalence and incidence of AFib is increasing and projected to double between 2010 and 2030, and that correlates with aging of the population and our obesity epidemic. There are an estimated 50 million individuals worldwide with atrial fibrillation estimated, that was an estimate from 2020, and at least 5.6 million in the US as of 2015. And about 11% of cases are estimated to be undiagnosed. The overall lifetime risk is 30 to 40% in white individuals, 20% in African American individuals, and 15% in Chinese individuals. And then if this statistic is old, but actually very interesting, and was pretty striking to me. And Medicare beneficiaries, so over 65, the most frequent outcome in 5 years after diagnosis is death. So look at these numbers, 19.5% at 1 year and 48.8% at 5 years. That's like an ICD population. So it's really a remarkable statistics. And probably because AF is associated with increased risk of many other things, like the 1.5 to 2-fold increased risk of death, 2.4-fold increased risk of stroke, 1.5-fold risk of cognitive impairment and dementia, and 1.5-fold increased risk of MI, 2-fold risk of sudden cardiac death. And especially noted recently with a Danish study, a 5-fold increased risk of heart failure. That's even higher risk than stroke. So it accounts for billions of dollars of healthcare spending. So it is our most common sustained arrhythmia. And it's associated with stroke, as you know, atrial cardiomyopathy and heart failure. And what was striking to me is that it's associated with very heterogeneous cardiovascular stresses like older age, obesity, hypertension, diabetes, coronary disease, valve disease, and heart failure. You can see the striking increase of AFib prevalence by age in these two studies. So going back to our understanding of AFib, from the 1950s to 1990s, we understood that AF is a reentrant arrhythmia. There's a multiple wavelet hypothesis that was put forth by Gordon Moe and his colleagues in the 1950s, where you had multiple wavelets of electrical activity occurring in the atria that lead to atrial fibrillation, and then structural substrates. So the larger the atria you have, the more of these reentrant circuits you would have, and then the more likely you would be to sustain it. Then in 1998, so about 27 years ago, there was a very pivotal paper that was published by Michel Hasseguer and colleagues in Bordeaux, where they reported that you could have focal mechanisms, that you could have AFib that was triggered and originated from the pulmonary veins, so where you would see these pulmonary vein potentials that could initiate atrial fibrillation. That has been paradigm changing, as you know. And so we changed our understanding of AFib so that now we have an importance of triggers coming from the pulmonary veins, and about 95% of them in that paper, these triggers came from the pulmonary veins. And in people who are susceptible, these triggers come out and conduct to the atrium and develop atrial fibrillation into this rhythm that we see with these pulmonary vein potentials and atrial fibrillation that may or may not sustain. So along with that came this dual substrate model, where for initiation and maintenance of AFib, you have the substrate for atrial fibrillation initiation in the pulmonary veins and the substrate for AF maintenance. And you could have electrical remodeling, structural remodeling, and have different contributions of each. And a lot of this is is contributed to by some of these risk factors like stretch of the atrium, calcium overload, LVH, LV dysfunction, valve disease, autonomic tone, inflammation, fibrosis, and toxins. So this dual substrate model kind of went along with what we see as the natural history of AFib, where people may start just with having frequent atrial ectopy, and these are probably representing the sources of AF initiation for the pulmonary veins. And they may develop along the years into getting repetitive atrial tachycardias, paroxysms of AFib. Then this could develop in a significant number of people into more persistent forms of AFib and even permanent atrial fibrillation. And as you go along this natural history, then the substrates for AF maintenance become more and more important. So along with this, over the last quarter of a century, we've developed atrial fibrillation ablation techniques. So the cornerstone of AF ablation now is pulmonary vein isolation. Now, before we knew about these triggers, there was the Cox-Maze procedure. So this was done in probably the early 1990s and still is done today with pretty high success rates, where Jimmy Cox at Washington University, where I trained, created this operation where he did slices and incisions throughout the atria to try to limit the reentrant circuits. And I remember sitting in a lecture hall, an auditorium where Jimmy Cox was doing a live case. And he'd been an arrhythmia surgeon, so he was doing a lot of AVNRT and WPW surgery at the time. And he developed this atrial fibrillation surgery. I think it was inspired by his mother, I think, who had atrial fibrillation and a stroke. And I remember him saying, "This is one procedure those damn electrophysiologists are never going to do with the catheter." So I thought that was really pretty funny given what then happened. We had John Schwartz, who developed kind of a maze, catheter maze a few years later. And I think we know now that the reason why it was very successful, one of the reasons, is that there was a box lesion around the pulmonary veins. So he actually did pulmonary vein isolation. So we've evolved from that to doing it with a catheter. And we have various forms of isolating the pulmonary veins with radiofrequency energy around each of the pulmonary veins, a cryo balloon where we can isolate pulmonary veins. And then more recently, over the last year in the US, we have availability of catheters that can do pulse field ablation, so like high voltage electrical stimulation that causes electroporation of the cells. And this has been really quite transformative, as you know, and the EP group knows here, really a huge amount of growth for us in EP because this increases the safety of the procedure. But there's still some remaining issues in AFib ablation. There's a ceiling of success, even with paroxysmal AFib and even with pulse field ablation. So the duration, the durability of the lesions is not 100%. And even in lone AFib, you know, our success rates may not be higher than 80 to 90%, even, you know, and that's counting some redo ablations. And so we see recurrence after successful pulmonary vein isolation. We see recovery of conduction sometimes across the lines, and new atypical atrial flutters can happen. We have not great success rates with persistent AF. Now, whether or not PFA is going to change that or not, it's possible. So we'll wait and see for some of the studies coming out. And the role of substrate ablation, ablating outside of the pulmonary veins to the other triggers that may develop as the disease progresses, is unclear. You know, what should we actually be ablating? We also want to reduce the complication rates. To some extent, PFA has really reduced a lot of these feared complications of pulmonary vein stenosis, atrioesophageal fistula from ablating in the back of the heart posteriorly, the atrial. We still have issues with atypical flutters. And then some new risks of hemolysis and coronary spasm that we can see with pulse field ablation, although it doesn't seem to be quite as clinically prevalent as we fear. But because of all these ceilings of success and the issues with AF ablation and limited some of these complications, early on when for the last couple of decades, we were asking in our group, are there upstream therapies that can actually target AFib substrates? Because AF was not like WPW. We can go in and cut one one pathway. It was pretty obvious early on that you couldn't do just pulmonary vein isolation or go in and ablate the focus that you map in a pulmonary vein. First of all, you get pulmonary vein stenosis. It's kind of a finicky little area. You had to ablate all 4 pulmonary vein ostia in order to get better success. So it sort of indicates there was like a more systemic thing going on. So early on, we started thinking about looking at upstream therapies for AF, looking at the inflammatory pathways, fibrosis pathways, calcium overload, and oxidative stress. And I collaborate with a close colleague of mine, David Van Wagener, who's a basic scientist. So people were targeting statins, steroids, ACE inhibitors, ARBs, and looking at some secondary analyses of randomized studies using these. And although they looked promising, what was disappointing that with the exception of steroids, which you can't take long term, or statins for post-op AF, and ACE inhibitors and ARBs and heart failure, when you looked at randomized clinical trials targeting AF, the results were really quite disappointing. So then in the 2000s, in that era, we started learning that AF can be more heritable than we thought. So we'd always had familial AF, and we knew that there were families that had high susceptibility to AF. But what we began to understand was that with common AF, that it can be heritable as well. So there's some data coming out of Framingham where parental AF increased the risk of AF in offspring. If you have a parent with AF, the odds ratio is 1.85. And if you had a parent who had AF early onset, then the odds ratio was over 3. And then Iceland with Decode Genetics, they reported similar findings that AF increases the risk of AF in first-degree relatives with a relative risk of 1.77, and it was the relative risk was almost 5 if you had early onset AF below age 60. And then at MGH, Patrick Eleanor and his colleagues showed that lone AF really increases the risk of AF in family members, and that's AF in the absence of other structural heart disease. And look at these relative risks, like sons had a risk of 8.1, daughters 9.5, brothers 70, sisters 34. And if you take a careful family history, he found that 30% of people with lone AF have a family history of AF. There's a Danish twin study that reported that AF heritability was 62%, and then UK Biobank 22%. So perhaps genetics is the ultimate upstream approach. And indeed, in 2007, Decode Genetics, which had genotyped about 100,000 of their population, reported the first genome-wide association study in AF. So what happened around this time was that the genome was sequenced. And what you can do is that you can have these assays that test for hundreds of thousands, now millions of markers across the DNA variants called single nucleotide polymorphisms. And what you do is these huge case control studies where you have a set of people with a disease or condition and a control population, and you do these assays. And it was pretty expensive back then. It was about $500 per sample. But you could do these big case control studies. And for the first time, the patient could tell you what's going on with AFib. Like before that, if you think about our antiarrhythmic drugs, they're like targeting ion channels because AF is an electrical disease, right? So, but for the first time, we could in an unbiased way, look across the DNA and say, which of these areas are associated with AFib, and which of these SNPs are associated with AF. So Decode Genetics reported this site on chromosome 4, Q25. They replicated in a couple of cohorts. And I'll show you that at the same time, we were gathering a lone AFib gene bank at the Cleveland Clinic and trying to gather enough samples to be able to do these GWASs. So this is called a Manhattan plot. We had about 500 cases of lone AF, and we had about 3,000 control samples. And this Manhattan plot, along the x-axis is each chromosome, and each of the dots is one of these markers, a single nucleotide polymorphism or SNP. And the y-axis is negative log of the p-value. Now, because you're looking across hundreds of thousands of SNPs, you have this big multiple testing problem, right? So you have to account for that. Instead of having a p less than 0.05, you have to adjust for multiple testing because 5% would be really a lot of false positives. So this line here is Bonferroni correction, where in order to get genome-wide significance, you really need a p-value of less than 10 to the minus 6 to 10 to the minus 8. So below that's a lot of noise here. And what you see here is this really strong locus at chromosome 4. And you could see this in our study with 100 patients. This is like the poster child of genome-wide association studies was age-related macular degeneration, and they could see a signal with 100 patients. And you can see this with AFib as well. So we didn't actually publish this beautiful Manhattan plot. Doesn't really look like Manhattan, but we contributed it to a consortium that was sponsored by NHLBI called the CHARGE Consortium. And it's cohorts for heart and aging research in genomic epidemiology. So we contributed our data to that consortium, and about 10 years later, we have 18,000 cases and reported 24 AF GWAS loci. So this now is starting to look a little bit like Manhattan. And then in 2018, the next year, there were over 80,000 cases, and then with over 100 AF GWAS loci. And then just this year, with over 180,000 cases, there are over 350 AF GWAS loci. We're at like, on some of these, p-values less than 10 to the minus 500, with this 4q25 locus. And then some other loci are showing up as well. So what's going on at 4q25? Well, it turns out that it's in a non-coding region, and the nearest gene is called PIDX2. PIDX2C is the cardiac isoform. And it so happens that along at the same time, there's a Dutch group that was doing a lot of work on PIDX2. And it happens to be a transcription factor that's active early in development, and it's involved with left-right formation of the heart. And not only that, it's involved with formation of the pulmonary veins. So out of all the genes out there, we land at a locus that's near a gene that's involved with formation of the pulmonary veins. We thought, wow, we hit, it's like eureka time. It's expressed in the human and mouse left atria and essentially absent in other parts of the heart. And it appears to suppress a left atrial sinus node program. So maybe that's what's kind of happening within the pulmonary vein. And there's Jim Martin especially has done a lot of work. He's at Baylor. He's done a lot of work on PIDX2 deficient mice. And he showed that they're more susceptible to pacing-induced atrial arrhythmias, and they don't develop a normal pulmonary vein sleeve. There are a lot of complex developmental transcription factor interactions. And also the other thing, it may have a role in early cardiac regeneration and repair. So it's really interesting. I thought early on, wow, this is what we should do. We can test for 4q25, and then I'll choose the people that we should ablate, and isolate their pulmonary veins, because could this explain the pulmonary vein triggers? So as Edwin and other electrophysiologists know, that when we do pulmonary vein isolation, we can record some of these pulmonary vein triggers. This is an ECG up here of a patient in atrial fibrillation, and this is recording within the pulmonary veins after we've electrically isolated. And you could see the firing going on within the pulmonary veins. And this is a patient who is in sinus rhythm, and you see this dissociated, what we call dissociated firing. So indeed, more lately what we've done is we've looked at there's this technique called spatial RNA sequencing, where you can actually look at across some tissues and do RNA sequencing to look at gene expression within areas and trying to get down to a cellular level of where these genes are expressed. And so this is a small study, but we were looking at some of the important genes that we think are involved with AFib, including PIDX2. And you can see in some of the yellow that it is expressed in these left atrial tissues. We got some donor tissues, transplant donor tissues of rejected hearts, where you could look at the slices of tissue, the junction between the pulmonary veins and the left atrium, and also the left atrial appendage. So we see PIDX2 being actually expressed in these. And also, what I thought was really interesting is HCN4, which is a pacemaker current, that we actually see some expression there. It's not real high, but you actually see some expression there. So maybe it does cause some of that. So so we thought maybe we could use some of these markers to do gene-based interventional therapies. Would it mark people who who would respond to pulmonary vein isolation. Well, the opposite was true. One of the first things, I thought, well, okay, so another hypothesis shot down. but it's actually very interesting because it turns out that the top loci associated with AFib, actually instead of marking successful ablation, actually it's associated with higher risk of atrial fibrillation recurring. And so there are conflicting results, but when you actually look at some of these, especially a study by Ben Shoemaker that was AFib ablato-genetics trying to do GWAS or limited GWAS of that, it's actually associated with higher risk. So, it kind of goes along with something systemic going on, maybe even beyond the pulmonary veins. So one of the things that was intriguing to me was this work looking at PIDX2 and cardiac regeneration. This story is not finished yet, but Jim Martin at Baylor, as I mentioned, is looking at this PIDX2 deficiency in mice. And it turns out that if you have neonatal mice and you lop off their apex of the heart, it regenerates. But in PIDX2 deficient mice, it doesn't regenerate. So he's also looking at that in an MI model as well. So he's studying these signaling pathways that are associated with regeneration, and it turns out that PIDX2 interact with some of these pathways, which is really interesting. Now, I haven't figured it out in terms of the directionality. What I don't know is whether or not after AF ablation are we reactivating PIDX2. We don't know, right? So it's hard to get those tissues. Right? So anyway, but this could potentially explain maybe why there's more recovery and reconnections after PVI. So the next thing that we were really intrigued by is the association of AFib with structural heart disease. And I think that's becoming more and more recognized lately, especially with some of these reports, the Danish study looking at this 5-fold increased risk of AFib with heart failure. And it turns out if you look at the genome-wide association study for heart failure, this is another Manhattan plot now looking at heart failure, no heart failure, there's actually overlap of some of the genes or SNPs that cause AFib and heart failure. So here's our friend here, our locus on chromosome 4q25. It's like one of the strongest loci associated with heart failure, this PIDX2 nearby locus. And there are some other areas, some other genes that have been associated with AFib as well that are overlapping with heart failure. And if you look at a clustering analysis of genes that have been prioritized and associated with AFib and look at the pathways that is involved with, you see a lot of structural genes. So here's some ion channel binding clustering, but really a lot of these are very much structural genes in the cytoskeleton or elsewhere. So, this is something we're learning from our patients because we never really knew any of this. Again, we thought that it was all very ion channel based. So this, leads to is in support for some of the screening that we're now, genetic screening we're now recommending from the AF guidelines, where we're recommending that early onset AFib, you should consider sending for genetic counseling and screening. Because if you look at, how many of these loci that you have common and familial AF forms that are associated with certain genetic loci, and some of them are structural genes, like Titin has been reported in these GWASs. It's a big gene, it's a big structural gene, and some of those loss of function variants are associated with AF. And a lot of other genes that are associated with inherited cardiomyopathy have been associated with AFib as well, like Titin, myosin heavy chain genes, and lamin A, and then this is a KCNQ1, it's an ion channel. But, and then we also know that a lot of these disease-associated genetic variants are more prevalent if you have a younger age of onset of AFib. And if you do sequencing, looking for the rare pathogenic genetic variants, not the common variants, then a lot of them are in structural proteins, and they may play a role in early onset AFib. So there's some studies, clinical studies from TopMed, where if you look at there were 1,293 patients with onset of AFib 65 or younger, and 10.1% of those had a disease-associated genetic variant in genes associated with cardiomyopathy or channelopathy. And if you look, there's a smaller study of with age of onset AF under age 45, 23 patients, and no identifiable cause of AF. 24% had a pathogenic or likely pathogenic variant, most in cardiomyopathy genes. So if you look at the genes where these rare coding variants have been implicated in early onset AFib and how they overlap with cardiomyopathy or inherited arrhythmia syndromes, again, here you see a lot of structural genes. So this has given a lot of support to this now 2B recommendation from our AF guidelines, where if you have AFib with onset of AF before 45 years of age and no other obvious risk factors for AF, that you might want to consider referring them for genetic counseling and genetic testing for these rare pathogenic variants. And what do you do with it? Well, that's what I was asking. You know, once you see something, I think you just may increase your surveillance for development of cardiomyopathy. So just to kind of summarize some of our perspectives so far from AF genetics and genomics, we have top AF locus, that's near a gene implicated in formation of the pulmonary veins, and that these pulmonary veins are the target of our AF ablation. The top risk loci appear to predict tendency for AF recurrence after ablation, and then there's overlap of AF and structural heart disease. Now this brings up, if these developmental genes are associated with AF, why don't we get it until 5 to 7 decades later? So that was like something that was really confusing to us. So what we've done in our group is to try to take to try to understand things stepwise. So we're following this paradigm, genes to function. Even though you've identified some of these GWAS loci, it's a really long pathway to trying to figure out how they actually cause AFib. So we have access to a big an AF tissue bank, an atrial tissue bank that David Van Wagener started several decades ago. And so we've accumulated over 1,000 left atrial appendage tissues, for example. So we can then look at how the genes are transcribed. You know, how the mRNA, we can follow this genes to function paradigm, looking at DNA to mRNA to protein, translated to protein, and then to function. So when you identify the SNP, the variant, that doesn't necessarily mean you know what the gene is that's associated with that it's acting on. So what you need, the first step is to try to figure out what those functional links are, what gene are you talking about. And then look at how gene expression is affected by AF, and then again, try to figure out what the causal SNPs are, because those SNPs that you identify may be in linkage disequilibrium with actually the causal SNPs. And then that may be, you don't exactly know what the actual gene target is. And, so we wanted to see if we can find new potential upstream therapies, because developmental genes are really tough targets. Like, you're not going to go do gene engineering, in a fetus or a baby, right? So what can we target in adulthood? So the first question we asked was, what are the causative SNPs and what are the genes that are affected by it? So we could use our human left atrial tissues and look at their genetics and look at that mRNA. So you can now do mRNA sequencing across the genome and you treat mRNA as a quantitative trait, and you look at the genotype and see how it predicts mRNA expression, and that could tell you which genes are involved. So that's called cis expression quantitative trait loci. So for example, you have a certain allele, and here's the G allele versus C allele. The G allele acts on this gene, and you get lots of copies of mRNA versus a C allele causes only a few copies. So you could see that probably is causative. So that's what we tried to do in our tissues. And a number of years ago, we published that in 2017. We published that in this current GWAS at that time that had 24 AF GWAS loci on that GWAS. We found significant EQTLs for 12. And so a lot of those are in blue and red. And it wasn't necessarily the closest gene because you can act from afar. But if you'll notice on this Manhattan plot, unfortunately, PIDX2 is in black, so we thought, oh, this is going to be easy. We're going to find that it's associated, that the 4q25 is acting on PIDX2. And the one that we wanted to find an association with, we couldn't. So why is that? We think it's actually timing, because we're looking at adult atrial tissues, and this is a gene that's active early in development, and then maybe location. So we're looking at the left atrial appendage, but not necessarily within the left atrial pulmonary vein junction. They're near each other, but not necessarily exactly the same. We are trying to do this kind of work in the pulmonary vein left atrial junction. Here we've taken one of these donor atria and like cut it up and tried to take different sections. And there are some small associations, but it's not really marked. So but on the other hand, kind of your left atrial appendage is like a barometer of what's going on in aging in human adults. So again, we're now going back to trying to figure out this age paradox. What happens to gene expression in AFib? So these are heat maps. We took our left atrial tissue samples and we could divide them up into whether or not they had AFib or not. So we had 3 states where we had control patients that never had AFib going for cardiac surgery. They have their atrial appendages taken off, and we call them sinus rhythm. We have people who are paroxysmal, who have a history of AFib, but they're in sinus rhythm at the time of tissue collection. And then we have the people with persistent AF. They have a history of AF, and they're in AF at the time. So if you do comparisons, you can make some inferences between AF susceptibility and then progression of AF if you're looking at prediction of development of persistent AF. And what I want to show here is that if you are in AF itself, you get a lot of changes in your genes just from being in AF. So it's really a complicated analysis. It's not so easy because you're not necessarily, AF, just being in AF itself really causes a lot of changes. And then as you can see, you get this alphabet soup of genes. And, you don't know what what a lot of these genes do. So you can do what's called gene set testing, which it analyzes groups of genes, which may be a little more robust than analyzing single genes alone. So you could do pathway analyses, et cetera. And what we found was that when we were looking at the susceptibility and persistent analyses, my hypothesis was, this is not going to be all that exciting. We've been working on inflammation all these years. We're just going to see an increase in inflammation and oxidative stress pathways. But to my surprise, instead, what we saw was like a decrease in some of these stress pathways with AFib. And so with susceptibility, there was a decrease in transcriptional response to these cell stress unfolded protein responses. So one of the areas that I thought was really interesting were these heat shock factor targets and ATF targets, which looks at this pathway called heat shock unfolded protein response. So it turns out that this heat shock response is a system that's designed to protect our cells from stress. And it ensures that your proteins are folded appropriately. And in fact, one of the hallmarks of aging is loss of this proteostasis. And heat shock factor 1 is the master regulator of this heat shock response in mammals. So what happens is when our cells are exposed to stress, like heat shock, endoplasmic reticulum stress, or oxidative stress, your folded proteins may actually unfold. And the way your cells take care of it is either by autophagy, by eating it back up, or by degrading it in a proteasome, or chaperone-mediated unfolding where it refolds the proteins. And heat shock factors are associated with that. But if the system is overwhelmed, what happens is that the unfolded proteins kind of aggregate and they precipitate, and that's a hallmark of aging. So they precipitate as amyloid. And you know all about amyloid, but it turns out that if you look at the atria, that atrial amyloid's been reported in over 90% of persons over age 90. And it's been associated with deposits of atrionatriuretic peptide, which is very highly expressed in the left atrial appendage, really highly expressed. And you might notice this in your patients. When they go into AFib, if you ask them, some of them say, I have to urinate a lot. They're like releasing ANP. So and this amyloid, as you know, can be proteotoxic and do a lot of bad things. We actually did a small pilot study looking at our left atrial appendage samples. We matched them, people with a history of AFib and not, and matched them for sex, age, and BMI, and did Congo red staining. And we saw indeed that there was a higher amount of Congo red staining in our AF patients, and it was not associated in our matched cohort with age, sex, hypertension, or BMI. So we're trying to follow up with this. Now we have like 250 samples that we're actively looking at. The other thing we've done with our mRNA expression is to look at how they interact with other genes. So we do what's called co-expression. How do those genes co-express with other genes? And it might highlight some other genes that are involved with AF. And Sojin Was, who's a fellow with us now, she's now an EP fellow, did a lot of work on analyzing these pathways. And if you look at this, the top pathways that are associated with AF have to do with some metabolic pathways, with oxidative phosphorylation, mitochondrial dysfunction. There's the structural signaling genes, and also some other stress response pathways. So what we have now described is this dual risk model, sort of the perfect storm for AF, where in genetically susceptible people, that inadequate transcriptional responses to stress may predispose to AF later in life. So you may be born with genetic susceptibility that, like the PIDX2 locus and SNPs that put you at risk for developing pulmonary vein triggers. Those are genes that are active in development. But it may take a second hit or multiple hits, like just aging, which is, I can tell you, causes a lot of stress, and obesity. Those are big stressors. And as we develop in life, that these stressors may overwhelm the transcriptional response to cell stress and lead to atrial fibrillation. So this brings up the potential for novel upstream therapies. We now have a lot of candidate pathways. We have candidate genes. And one of the genes that we're chasing after was called FAM13B. There is not a lot. This is one of the hits on chromosome 5. And we knocked it down, and this is done by Jonathan Smith, another one of our molecular biology collaborators. And we looked at pathways that were knocked down when we knocked down FAM13B, or what was changed with the knockdown. And we saw at that time that it was associated with some changes in some mitochondrial pathways, where these are associated with mitophagy. It's like autophagy for proteins, except that as we age, we develop more dysfunctional mitochondria, and these get taken care of by getting eaten up by some of these genes. So that led us toward looking at whether or not this is a process that is involved with aging and an AFib, and it led us to this enzyme called AMP kinase. And it turns out that AMP kinase is the master regulator of metabolic stress in the cells. It looks at AMP-ATP ratios, and in response to stress, and it can increase ATP generation and decrease ATP expenditure. And it turns out that certain factors are associated with changes in AMP kinase, including caloric restriction and exercise. And we do know from our colleagues in Australia that lifestyle changes like weight loss and exercise are associated with a decrease in AF burden and decrease in AF. And it's led to a lot of our recommendations nowadays for lifestyle risk factor modification. And also, very common drug, metformin, has activity on AMP kinase. So that led us to testing metformin and lifestyle risk factor modification. And we looked at our co-expression analyses that also support that some of the it implicates some of these subunits of AMP kinase, this AF being associated with decreased transcriptional response to stress. So with that, we were trying to go from basic lab back to the bedside by targeting this novel upstream therapy for AF, these genomic targets. So metformin, as you know, it's a very cheap first-line therapy drug for diabetes and prediabetes. It works in the mitochondria with downstream activation of AMP kinase, and it lowers reactive oxygen species production, increases cardiomyocyte ATP production via AMP kinase. It promotes mitophagy to clear these dysfunctional mitochondria. It promotes weight loss, and it's being studied for its potential anti-aging effects. It's been used off-label in studies without hypoglycemia, and there was an observational study in Taiwan where it was associated with 20% less AF in diabetics. We took the metformin in vitro first to look at its response of our beating cardiomyocyte cells derived from inducible pluripotent stem cells to look at some gene expression. So we looked at gene expression of some heat shock factors, and they seem to go the right way with or without pacing stress. and also some calcium channel gene expression and sodium channel gene expression seemed to go the right way. So we designed this study called TRIM-AF, Targeting Risk Interventions in Metformin for Atrial Fibrillation. So the study design was it's a prospective, randomized, open-label, blinded endpoint probe, 2x2 design, where we took people who have implanted pacemakers or defibrillators. So they have a lead actually sitting in their atrium, and they can give us daily AF burden and daily activity, actually. And it's a 2x2 trial. We intended to enroll 200 patients, and there are four arms: control, metformin, lifestyle risk factor modification, and lifestyle plus metformin. So unfortunately, we did this during COVID. COVID happened right as we were ramping up, which makes it really hard to do a lifestyle study, I have to say. And we had to cut the sample size down to 150. And at AHA this past year, we reported the first results from the first year, from year one. And unfortunately, what we actually saw was that metformin trended worse than the standard of care. And actually, our standard of care, we realized it actually did some of the best reduction in AF burden. AF burden went down on all of our arms. So there's a lot to unpack in our study. But it turns out we probably did some lifestyle risk factor modification counseling just in enrolling people. And we gave them a bunch of handouts. We're now calling them the magic handouts because they actually reduced their AF burden a lot. So it's kind of hard to beat standard of care compared to lifestyle risk factor modification, where we were having them do very frequent contact with our preventive cardiology group. They would see a dietitian. They would do they saw exercise physiologist, and had a lot more intervention. And the problem with metformin, I think, was that we didn't realize was that it has a lot of side effects to it. And it turns out that half of the people could not tolerate it, and they weren't on it at 1 year. So, and they had, all those GI side effects, and I just wonder if we're increasing their inflammatory load, doing something to their microbiome that is harming AFib. Now, we still have a 2-year endpoint coming up, so we're still, it's still continuing. And the other thing we've done, though, with this clinical trial is that this was not a pragmatic trial. If I had to do another trial, we talked about GLP-1 RAs, and this, I would do a more pragmatic trial. We actually did this to try to collect a lot of data, a lot of samples. We actually were thinking that we could personalize AF treatments, and we collected their blood samples, did genotyping on everyone, and we cryopreserved their white cell counts. So now there's another development in our field where Professor Yamanaka published that you could incubate fibroblasts or skin cells, and now even white blood cells from just a blood sample with 4 transcription factors, and reprogram them into stem cells, inducible pluripotent stem cells. And then you can then differentiate them into various other cells like adipocytes or hepatocytes. And we differentiate them into beating atrial cardiomyocytes, and you could do ventricular as well. So the thinking is that you could actually take a person's blood samples, and you can't do multiple testing on heart biopsies, right? So, but you could actually make cardiomyocytes from patients, and they express PIDX2 and all this, and you could have this model in a test tube in a petri dish, and they actually form syncytia, and it's pretty impressive. Actually, the first time I saw this, you could see that they actually beat so much. You could see ripples of activity and contraction going across it. So it's actually a problem. You see in this figure here that they're actually pulling off the plate, so it makes it a little bit hard to actually measure them. But it's a pretty cool system because we then grow them into engineered heart tissues. We grow them between these flexible posts so that they're kind of exercising, and it helps them to mature more. And you can kind of, they move. I don't know if there's a video that can show here, but they actually move and they mature more. You can look at their transcriptomics and see if they go, you can test drugs. So here we've just given, it's not working. But anyway, you can give them isoproterenol, and they beat faster. You can do optical mapping of the conduction across these engineered heart tissues. So now we have a way of potentially testing drugs in vitro. So this would be like one of the first times that it would be done in a cardiovascular system, because they can do this a little bit in cancer tissues, but we haven't really been doing this so much in cardiovascular disease. So we're kind of looking at ways to go from these genes to drugs. So one way that we're doing this now is to actually use network systems biology approaches to trying to repurpose drugs and then hopefully identify new drugs. Because it's reported that genetically supported targets can double the clinical development success rates. And we use network medicine where we incorporate different layers of data that's related to biology, environment, to try to understand these complex diseases. We identify disease networks and then identify drug targets. So the way this works is that, and this is with Dr. Feizhong Chen, who is now heading some of our genetics at Cleveland Clinic, where you could take the human interactome, the protein-protein interactome, and various other protein-protein interactomes, and develop a disease module. So this is like one of our first AF disease modules. And then you have drug databases where you know where they've been studied before, and you can look at how close they are in terms of targeting genes in your disease network. So you do this proximity analysis. So we published this. Dr. Jessica Lau, who was a graduate student, did the first study for us where we integrated our transcriptomic data from our engineered heart tissues, our atrial tissue actually in this case, and layered it upon some of the protein-protein interactome data. And this is our AF gene disease network. And we did proximity analysis and identified 55 top drug candidates. Metformin is on here as well. And what we try to do is we try to see whether or not those drugs act in a different direction in terms of gene expression to what we see in our atrial tissues that I showed you with those heat maps. Like, do they go towards a more sinus rhythm look? So here you have, this is AF gene expression of these top genes that were changed with a drug called dantrolene. And dantrolene goes in the opposite direction from red and blue in an opposite direction to what we see. So this was another target that was on one of those networks. So, but we're going to be more careful this time, because what happens is that you don't know which direction it's going. It could be harmful or not. Okay, so you just know that the drugs target a gene. Okay, and in fact, here we know that dantrolene, unfortunately, even though it has some really interesting targets, like it's being studied by the Vanderbilt group as a potential drug in a variance of these drugs for ventricular arrhythmias, it stabilizes ryanodine receptor. But it has some significant side effects, so you're going to have to, you may be able to use some of these drugs, but then design something else, like we might have to for metformin. So this is a Gen 2 AF disease module. We have some others, and again, this highlights that some of these may be actually making things worse, like some of these theophylline-related things. And so this is what we're going after now, is looking at some of these genes, and we're looking at functionally testing them in vitro in patients, from cells that we're taking from patients who are in our TRIM study, so they have a history of AF, and then testing these drugs in those cell models. And then if they survive that testing, then we'll take them into some spontaneous AF mouse models. So I'm just going to stop here, running a little late, but just to summarize, that going from bedside to bench to bedside, these large AF GWASs implicate over 100 AF GWAS loci. The top locus is near a gene involved with pulmonary vein formation, and transcriptomic studies suggest that AF is associated with impaired or overwhelmed responses to cell stress. Our dual risk model proposes that in genetically susceptible individuals, inadequate transcriptional responses to stress predispose to AF later in life. And hopefully, with all this, we'll find with our multi-omics studies and drug targeting studies, we're still hoping to just find new drugs for AF, because there hasn't been a new AF drug in over a decade. So hopefully we'll get there. So thank you very much, and I want to thank my team that's been working on this.
Thank you, Dr. Chung, for a wonderful presentation. We're going to move on to the question section. And if anyone had any doubt that the basic science and genomics of AF is a complex endeavor. I'm sure we're all convinced right now. But similarly, the clinical presentation is complex. Some patients are very symptomatic. They have a few seconds of AF. They're calling EMS. Some patients are minimally symptomatic. Some patients develop rate-related cardiomyopathy. Some patients, their heart rate is well controlled and they get cardiomyopathy. Some patients get significant atrial dilation and atrial functional AV valve regurgitation and such. Do you have any insights as to genetic markers as to, if you can predict?
Heart disease markers, except that this is an area that is also another area of interest. So at HRS, Heart Rhythm Society, we're going to be launching a research network. And one of the areas that I hope that we can focus on are these biorepositories that may help answer some of those questions. Like the heart failure, why do some people develop heart failure? Can we do a reversible heart failure GWAS? Because some people will have tachymedia cardiomyopathy or dysynchrony, electrical dysynchrony, and it reverses. But it sort of indicates that they're susceptible to some sort of trigger that drops their LV function, some stress that drops their LV function, and can we determine what those are from genetics? So that could be one area that we could look at. We could look at symptoms too. There are now these huge databases that if we could figure out if, from Charge or other consortia where you can answer potentially some of those questions with some of the genetic data that's already out there. So that would be, those are interesting questions, right?
Thank you, Mina. This was fantastic. It's fascinating to see the prevalence of pathogenic and likely pathogenic mutations in genes known to cause cardiomyopathy in specific population with AFib. Do you think that's because AFib is part of the spectrum of the expression that has been missed because we have not been typically screening patients or maybe focusing on AFib as part of the overarching clinical expression of the phenotype, or is it a specific predilection for atrial myocytes maybe, because that would affect how we screen family members? And just to follow up on that, have you done cardiac MRIs on these patients to see if they have a ventricular or atrial cardiomyopathic process? Because this has significant implication on their family members and on themselves, on the patient themselves.
That's great questions and great avenues for your research. Research, to do MRIs on everybody and to look at their ventricular function. I think there's a lot of interest, including in our group, looking at HFpEF as AFib as the manifestation of HFpEF, and to look at those would be a really nice avenue of research. We've looked at mainly atrial CTs and MRIs. We're collaborating with Rod Passman at Northwestern, who they have and Ben Shoemaker at Vanderbilt. They have very large collections of MRI as well. So it's something that we've genotyped a lot of those, and we're in the couple thousand. So it's possible, we should collaborate and see whether or not we should look at something in their ventricles, something that we've thought about. But yeah, 50% of people with AF may develop heart failure. 50% of people with heart failure develop AF. So there's got to be, and there's all the clinical data that suggests that the stroke risk, et cetera, is not necessarily temporally relatable back to a specific episode of AFib. So maybe it is the structural milieu that we're looking at. Yeah.
First of all, I could tell you personally that Dr. Burchell would have very much enjoyed your talk, having known him for years. But the second question I have is sort of out in left field. Why is it that we have pulmonary vein muscle bundles anyway? Why in nature are they there? You could imagine that every time the left atrium contracts, it prevents blood from going backwards and sort of funnels everything through the mitral valve front in a frontwards fashion. But if you think about it, now that lots of pulmonary veins have been isolated in antrums, nobody seems to talk about hemodynamic consequences of doing that.
That's a great thought. I've never thought about the pulmonary vein osteocontracting. I don't know if we have resolution enough with MRI or something to actually look at that. That's really interesting. What's also I guess pulmonary veins have to connect to the atria somehow. So there's muscle. These connections are really interesting because PIDX2 is expressed in the pulmonary veins, it's also expressed in outflow tract. So it's connecting there. So we see PVCs. So what I wanted to do for a long time is to do GWAS of those PVCs to see whether or not the same, the muscle sleeves that go up into the outflow tracts, or we see SVC also, connecting to the right atrium. We see sources from there, whether or not they're all related. If there's some genetic predisposition, maybe still through the 4Q25. But yeah, I mean, it's kind of, that's an interesting question. I hadn't really thought about, are we preventing backflow from into the pulmonary veins and the hemodynamics? I don't know that anybody's looked at that.
It might have a direct implication.
Yeah. Thank you.
Thank you. Excellent talk. So I'm curious about the part where there are overlapping genes between both atrial fibrillation and heart failure. So, and as you were saying that recently there has been a growing evidence about the interdependence of atrial fibrillation with an entity like heart failure with preserved ejection fraction. So I'm curious to know if the type of heart failure with those overlapping genes was actually identified, and if the genes have some sort of independent, or if there is evidence of independent association with both entities, or could it be that the genes are leading to one of those two entities, and that could subsequently develop the other entity?
Yeah, so that's a great question that I wanted to, that's part of what we want to answer, like with our HRS research network, one of the areas, because so far it's been separate, right? And they happen to overlap. But we haven't really looked at that reversible cardiomyopathy phenotype, where AF, and so I know in our team, I've been interested in this for a couple decades, so we've coded history, even though our initial gene bank was lone AF, we've coded for if they've ever had an LVF less than 50%. So I'm kind of hoping that we could get enough centers, or as well, to really join in that effort, where we could try to look at whether or not what the overlap really is between AF and heart failure genetics. That'd be really, and you might, it might be a good discovery thing, like if you find pathways, genes that you can target for reversibility. That'd be pretty cool, right?
Forever we called something ARVC until we figured out it's not R, it's arrhythmogenic cardiomyopathy, and can have different phenotype. Could this be part of the phenotypic expression that the atrium still has cardiomyocytes, and arrhythmogenic cardiomyopathy could be arrhythmogenic in the atrium? So it is fascinating. So.
Yeah.
Thank you all for attending. John, you were going to ask a question?
Yes, thank you, Dr. Chung, for your time this morning. We did have one virtual question that just came through the webinar. It comes from Dr. Sajati. And he's asking, are there associated changes in the microbiome with atrial fibrillation?
That is a great, so we have a program project grant and 4 projects in that. One is on the microbiome. So Bob Keith, who worked with Stan Hazen, who did a lot of work on the microbiome, and Bob actually published some of those papers on the microbiome in cardiovascular disease. So he's been looking for us for the microbiome in relationship with AFib. And he does see a lot of association with some of those. He's much more versed in talking about the factors in the microbiome, but some of the similar ones, and also looking at inhibitors of that. So that work is just starting, and I know that he's just submitted a paper. So yeah, I think there definitely are changes in the microbiome that we're going to look at a lot more. We did collect stool intrameaf. I told you it was not pragmatic. It was really difficult. So we have some. It was obviously it was optional, but so we can look at stool. And Bob is actually looking in mouse models, so and doing a lot of really elegant work in the microbiome. So I think we'll hear a lot more about that coming forth. Thanks for that question.
Thank you for coming in. I know we could go on forever.
Thank you.